Hi, my name is Vanessa, and tonight I'll be talking about the structure and function of a DNA and how it relates to the RNA. So right here, I have a DNA in its full form, otherwise known as a double helix. So, since we're going to be talking about the structure, let's focus on just one side, make it a little easy. And so for this side, we've got the 3' at top, which resides in the hydroxyl group, and we've got the 5' at the bottom, which resides in the phosphate group. So when we're thinking about a coding strand, we're going to be focusing on each nucleotide in a DNA strand. We're going to be actually focusing on the bases of the nucleotide in the DNA strand. So first off, what's a nucleotide? A nucleotide consists of a phosphate, a deoxyribose sugar, and a base. And I'm sorry, my hair's a mess. Okay, anyways. So when we're thinking about coding strand, think of the coding strand as like a cookbook that's saying what recipe is needed in order to produce a delicious meal. So this coding strand right here is saying what it needs in order to be complete. And when it's matched with its perfect opposite, it's full. So this opposite is great for the one that I just demonstrated because it's got the 5' at top and the 3' at the bottom. And at the base, it's got guanine, which is perfectly opposite of cytosine, and adenine, which is perfect for thymine, and so on. And the sequence continues. So now we're going to be talking about mRNA and how it relates to DNA. So in this situation, here's the mRNA strand. This one is going to be pairing. It's got the 5' at top and the 3' at the bottom. And this is going to pair with a DNA single strand, which has got the 3' at top and the 5' at the bottom. So when these two match, it's going to be just about the same as a DNA double helix, except instead of thymine on the RNA side, it's got uracil. So that's its only different unique characteristic. Other than that, it's pretty much the same. Oh, and also the ribose sugar at the center of each nucleotide on this strand. So that's basically the coding strand and how matching works. So now we're going to talk about mRNA and how it interacts with tRNA, which is transfer RNA. So when the two match, scientists, you or me, we focus on a 3 nucleotide sequence. So in this 3 nucleotide sequence, we've got guanine, cytosine, and adenine, matching with cytosine, guanine, and uracil. So when the two match together, when a 3 nucleotide sequence is matched, an amino acid is created. So in this situation, we're going to be focusing on the one with the 5' at top, and that's going to be guanine, cytosine, and adenine. So we're going to refer to our codon table, and we're going to look for a sequence of GCA. So I don't know if you can see, but GCA is right there, and that tells us that the amino acid which will result with that sequence starts with ALA. So we'll refer back to ours, and we're gonna look to see which amino acid is created. And what do you know? We got it. It's alanine, abbreviation ALA. And that's it. Thank you so much. Have a great night.